The project that was supported by the Laboratory Support Forum was a project that was to identify genes that were differentially expressed or differently expressed in the same population of cells that seemed very similar to us, but that one of these populations was depleted in lungs that had been exposed to high oxygen. So we really needed to know what these cells made, because that would tell us perhaps what was deficient in the lung. And so we actually isolated cells, these two populations of cells, and then we ran gene arrays on them, which meant that we looked for the expression of every gene expressed in these cells, and we compared them between two different populations of cells. And we found those that were the same and those that were different. We found that about 0.5% of all the cells, of all the genes made by cells, were differently expressed in these two populations. So they're incredibly very similar populations of cells, one with a few limited number of genes that were different. What we found was that this population of cell was not only missing in the mice that had high oxygen, but now we also saw that these cells actually made an antiviral gene that blocked viral replication. So now we had a lung that was deficient in a cell that prevented viruses from replicating in the lung. And so now what we wanted to do was take that information and move it forward by restoring and rescuing the phenotype. So what we found using an experimental mouse model is if we give the mice high oxygen at birth, much like you might see if you were a child born prematurely, and then you allow the mouse to grow up and you give it respiratory viruses, what we find is the mouse that was born into room air gets sick and then recovers. The mouse that gets virus at birth gets sicker and still may or may not recover as efficiently. And because of the identification of this cell population that's depleted in the mice, and the fact that these cells actually make an antiviral gene that blocks viral replication, we've now developed a tool with a colleague of ours in which you can deliver that gene back into the lung. And because of that, you can restore high-level expression of this gene, essentially replacing what that cell was supposed to have been producing. And in doing so, now we've actually rescued the mice, much like you do with a vaccine. The mice don't get as sick anymore. And so now we have a new potential candidate to study in children that are born prematurely and ask, is this same gene or this population of cells deficient? And if it is, can we use these kind of tools that we've started to develop in experimental models? Can we use that to treat the children in the future?